we've talked about breeding strategies as a form of genetic engineering. But we do, now that we know so much more about DNA and the human genome, which is all of the genes in humans, we've tried to map it, and lots of other genomes, we do genetic engineering a little more different and a little more detailed. And the basic idea is, what if we could cut genes out of an organism and put them into another one? So a quick example is, I like to use the examples of diabetics. People with diabetes do not produce the protein insulin. And so if we could help them by cutting a gene out of, let's say, me, who produces insulin just fine, and give it to a diabetic person, maybe their pancreas would start producing insulin. Now, it's not quite that easy, of course, because they are already grown, and they're not really making new genes that fast. But that's kind of like the holy grail of genetic engineering, is to be able to heal people of genetic diseases by giving them new genes. Well, let's talk about how we can do it right now. One of the things we do now is we use bacteria to produce things we need. We use bacteria. Okay? Now, most of you think of bacteria as disease-causing organisms, but remember, that's not all they do. There's 99.9999% of all bacteria are harmless. So we want to use bacteria to produce proteins. Why bacteria? Well, first of all, bacteria reproduce fast. Bacteria can reproduce their genes every 15 minutes. And so you go from 1 bacteria to 2 bacteria, to 4, to 8, to 16, to 32, to 64, and so on. So in a day or two, you have millions of bacteria. Number 2, they're small. You can grow bacteria in a very, very small spot, a whole lot of them, in a small place, because they're single-celled and they're tiny cells. Their DNA is easy to get at. Bacteria don't have a nucleus. Remember that bacteria, we use the term prokaryotic, which means that they don't have a nucleus, which means that their DNA is just out in their cytoplasm, and so it's easy to get at, and it's easy to put into them. And they're cheap. You can find bacteria everywhere. So here's the basic procedure. It is to do this. Remove a normal gene from human DNA and insert that gene into bacterial DNA. Now, that sounds all well and good, but there's a little bit of a process that we have to do in order to do that. It sounds simple, actually. And it kind of is. But remember, in order to remove a gene, we have to be able to cut it out. And so the first step is to cut out the gene. We have to cut the gene out of the human DNA. So we're going to use the example of insulin today. Okay? We want to produce insulin. Okay. So we have to cut the human DNA with something called a restriction enzyme. Restriction enzymes, and I'm going to pop up the rest of this a second. Restriction enzymes recognize specific DNA sequences and cut the DNA at those sites. So, for example, the restriction enzyme called EcoR1, and they're named after the organisms, bacteria specifically, that they were found in. The restriction enzyme called EcoR1 recognizes GATTC, and it will cut the DNA like this. Okay? It will cut the DNA like that. It will cut down through the sugar and phosphate side of the ladder, between the bases, AATT, and then down again to make a little stair-step fashion. So you get what's called a sticky end. Sticky end means that if you have another piece of DNA that ends in AATT here, that ends in AATT, it'll stick on there, because they'll combine, like here. So there are lots of different, hundreds of different restriction enzymes that recognize hundreds of different sequences, but you have to have a restriction enzyme to cut out the gene. So here's our DNA strand. Here's our gene we want. We have to take a restriction enzyme that'll cut on this side of the gene and on that side of the gene. Remember, the DNA goes on forever, pretty much in both directions. And so we cut the gene out that way. So we take a human cell and we remove its DNA and we cut out the gene for insulin. Now what we have to do is put it into bacteria. So we insert it into a different cell, and in this case the different cell is a bacterial cell. We could insert it into other cells too in different ways. Okay, how do we do that? Well, it's injected. We take this little piece of DNA, here it is. Here's our gene for insulin. It's been cut open, so you see that the white here is where it was cut. So the gene for insulin is right here. Bacteria have inside them genes called, their DNA sometimes occurs in a ring called a plasmid, in a ring. So if we take the same restriction enzyme and cut open the plasmid, okay, we take the same restriction enzyme and cut open the plasmid, what happens to the plasmid is the ring pops open, right? It's like cutting open a ring of anything. It just pops open like that. And on the ends of the ring, we have what are called sticky ends, because this TTAA is right here. And this AATT is right there. And on this DNA, we have sticky ends, AAT, AATT, and TTAA. So this side reads I can't write small enough, TT. So we cut open the ring, and in the ring, I'm going to erase the ink a second, and then right here, our ring is cut open. Here is our piece of DNA we cut the gene out of, and we just injected it. If we inject it into the bacteria, what will happen is this end will pair up with that end, and this end will pair up with that end, because A's pair with T's and T's with A's. And you see this arrow here, it'll stick right in to the ring. So the bacteria will take the new DNA as part of itself. And we call that recombinant DNA. Recombinant DNA. So now our bacteria cell, I'm going to draw a bacteria, has its little ring of DNA, but now it has a new gene stuck in it. And what do organisms do with genes? They use them to produce proteins. In the case of this bacteria, it's now going to produce the protein. And the protein we want here is insulin. So what we're going to do then is we're going to take this new bacteria and we're going to let it divide. We're going to feed it, and we're going to let it divide millions of times to make trillions of bacteria. And those bacteria are all going to produce insulin. We're going to put them in basically, we're going to have them in this giant vat, and we're going to produce insulin, and then we're going to take the insulin out, and we're going to put it in little vials, and we're going to give it to diabetics. There you go. If anybody in here is a diabetic or knows one, if you can get a hold of their insulin vial, right on the side of the vial, it says made with rDNA, recombinant DNA. It's made exactly in this way. So there are some other things we use bacteria to do. We use them to make human growth hormone also for people with issues. There are lots of issues human growth hormone is used for. One is called pituitary dwarfism, where they don't produce any growth hormone. People with blood disorder, where their blood doesn't clot, we use it to make blood factor 8, which is a kind of thing in your blood that helps it to stop bleeding when you cut yourself. So we have changed genes in this way. There are a couple, there's another term that goes along with this. If we put these genes into different, those bacteria then are also called transgenic. We've taken a gene from one organism and put it into another. Here are some other organisms we've done that to. Maybe you've seen this picture before. This is a plant called a tobacco plant. In the same method we just talked about, when this plant was a seed, they took and they inserted into it the gene to make the protein luciferase. Luciferase is the protein that makes fireflies glow. And so when this tobacco plant grew, it glows in the dark because it has the same gene in it that fireflies use to glow in the dark. We've also made plants to contain genes that produce insecticides. We've taken animals and we've given them to give them growth hormone in their genes to make them bigger and with less fat. So besides this tobacco plant, we've made other organisms transgenic by giving them genes from other organisms. And so that's one way we do genetic engineering nowadays.